thank you very much for giving me the opportunity to talk here today. It's very nice, especially also to talk after some nice modeling from the Nicodemi lab, where I used to collaborate a lot when I was with Stefan Mundlos. Actually, I have some slide here which contain some content of the lab. So what I'm going to talk today about is not only about 3D structure, but a lot about also gene regulation and chromatin states. So the title of the talk today is the regulatory, transcriptional, and phenotypical cost of non-coding mutation at the Pitx1 locus. In my lab, we are interested in basically how during development expression patterns of genes are formed. And I just give you here an example. This is a limb bud at E9.5 with in red here gene that is expressed all over this limb bud. But when the limb buds start to grow, the pattern will progressively be restricted to very specific cell type with very limited boundaries of expression. And we think that this is due to the complex interplay that happens at the level of regulatory landscape, where a gene at one point from a poised state during development is initiated, its transcription is initiated, maintained for a certain amount of time in specific cell types, and then progressively decommissioned and repressed so that it doesn't produce transcript anymore. And all of this is produced, of course, at the level of enhancer-promoter interactions between genes and promoters. And many things are happening to allow a gene to enter this initiation phase. It is first the activation of the enhancers, changes in the 3D structure that enables the promoters and the enhancer to communicate together. And the talk that the data that I'm going to present today, they are mostly dealing with what happens at early phase and also during the early phase of maintenance or the early initiation phase, as well as the maintenance phase of expression. And the first question that I'd like to answer before the end of the talk is, can we see during the process of activation partially active transcriptional state that result from initiation in vivo, which means genes where some of the enhancer are active, but the gene itself didn't manage to overcome its repression, so that the activation is not enough. And this would be a byproduct of the moment of activation. Then what happens when we remove one of the multiple enhancer that control this gene expression? Does that decreases only the expression amount of the gene, or that leads to a more complex effect where we have indeed cells which are partially active, not fully active, and accumulated in a tissue? And finally, I will terminate the talk talking about a process that is called endo-activation. It is when an enhancer of one gene ectopically contact this gene and ectopically activate this gene, leading to a gain of function and often disease. And we want to go further into describing this mechanism and why some cells can undergo this ectopic activation, why others cannot. All of the data of today happens at one model locus, that is Pitx1. So Pitx1 is important to make the difference between the arm and the leg. So during differentiation, you have here a forelimb, which produce the arm, and a hind limb that produce a leg. And both structures are embryonically very similar, but during development, they differentiate into something that is different, homologous, but different. You can see the leg as here the kneecap, or has this bone, the fibula, that is very thin and bowed in comparison to the homologous bone of the forelimb that is straight and pretty thick. And if we look at the expression of Pitx1, we see that it's expressed here in blue only in the leg. So Pitx1 controls the leg development, and when we remove Pitx1, we see that the leg here in this scheme change its identity towards something that looks more like an arm, with the lack of the kneecap and this very thick and straight fibula here. Pitx1 is controlled as many developmental genes. That's why it's a good model by many different enhancers. And you can see here 4 of them, but there are probably other one that so far we did not manage to identify. And one of these enhancers is very interesting, is the PEN enhancer, for pine limb enhancer, because it is a a very strong enhancer, but it's also weird because it can activate a reporter activity in the forelimb and in the hind limb. So in the hind limb, it is understandable because it can activate Pitx1 there, but in the forelimb, this activity need to be somehow repressed or kept away from Pitx1 to avoid an ectopic activation of that gene. And we'll come back to that at the end of the talk. But on the first part of the talk, what I'm really interested into will be to show you how the PEN enhancer alone can contribute to the activity of the entire regulatory landscape, not only to the activity of the Pitx1 gene. If you remove the PEN enhancer from a mouse, this is what you get. You get a little bit less expression of Pitx1. You can see here in the hind limb, this is again the expression. You can see it's a little less strong, but this slight decrease in expression is enough to produce a phenotype that is a clubfoot phenotype, where the mouse is basically dragging its leg behind it. And we wanted to get more information about all the process that go from really the fundamental enhancer promoter interaction to the phenotype here. So we use two approaches in parallel. The first one is a pretty straightforward single cell transcriptomic approach, where we dissected wild type forelimb where Pitx1 is not expressed, wild type hind limb where Pitx1 is strongly expressed. Then we dissected enhancer mutant, so PEN deleted hind limb, and full Pitx1 knockout hind limb as a control. All of this went through single cell RNA-seq. And I just give a short overview before I go further. So the overview is basically, if you just look at the cells we get all the cell type of the limb. And one cell type is particularly important. It's the mesenchyme cell type that is here, because it is the cell type where Pitx1 is found expressed. You can see here in brown, this means just that the gene is expressed, and you don't see it in other clusters of cells that have other cell type identity. So we went further and just cluster the cell type to have a bit more detail of where precisely Pitx1 is expressed in the hind limb. And you can see here many different cell types, right, in different colors. We have a progenitor cell type, like the proximal progenitor or the distal progenitor. We have connective tissue, which will give rise later on to the tendons. So we have tendon progenitor here. We have a lot of bone tissue, which is this late digit condensation or the proximal condensation that will really give rise to the bone. And finally, we have also this irregular connective tissue here that is a patterning tissue that will enable the muscle to enter the limb and to actually connect to the proper part. What you can see here is basically the same idea. We can see also here a little bit that Pitx1 is not expressed in all the cluster in the same intensity. You can see here that is a bit stronger expressed on the left side of the cluster. And this left here is basically the proximal side of the leg. So proximal is here the red line. And you can see here that the expression amount of Pitx1 is stronger in the red line, which are proximal cells. In distal cells, we have less expression, but also very high amount of cells here that do not express Pitx1 at all. So this is the first approach we use to understand what happens to the deletion of an enhancer. The second approach, which enables us to go more deeply into the chromatin biology of these cells, is the very simple integration of a cassette directly upstream of the gene Pitx1. We also do it actually for other genes in the lab. But when you do that, what you hope is that this cassette will be able to sense all the regulatory activities around all the enhancers and basically reproduce the pattern of expression of the gene it is next to it. So here Pitx1. So we expected from this integration that the GFP would mark all the cell where Pitx1 is active, and then be able to go further with fact sorting and other type of analysis. And in a second time, we would add on top of this sensor the deletion of the enhancer to really see how the cells that express this marker gene would be influenced by that. So I just show you here, this is basically the pattern of expression of the GFP. So you can see here with different technologies, we can really see all the pattern of expression where we expected Pitx1 to be expressed, and so the GFP. You can see here in transparency in the embryo, the pituitary gland, the mandible, the hind limb, as well as the genital tubercle, which are all tissue where Pitx1 is expected to be expressed. And this is something that when we fax the cells, we see very well. So just as a comparison, if you take the forelimb and you look at the GFP in this transgenic animal, you will see that there is no GFP expression at all in comparison to hind limb, where a lot of cells display expression of the GFP. So what we just did is fax sort the cell in the hind limb that is supposed to be expressing tissue that are not expressing the GFP, and the cell that are expressing the GFP. We produced an RNA-seq data set from that. And you can see here that cells that do not express the GFP do not express Pitx1. A cell that express the GFP express Pitx1 very abundant. So we think here that this approach of ours really enables to sort, even in a tissue that seems overwhelmingly expressing a gene, the particular cell where the gene is expressed. Then we can move towards more chromatin study. And so the first thing that we did was to look at A3K27 acetylation, so this active promoter and enhancer mark. And we saw here something that was pretty surprising and actually answering to one of the questions I had in the introduction before, is that in the cell that do not express Pitx1, so the GFP negative cells, we don't see any acetylation at the promoter, neither at any of the enhancers. And these tend to show that these cells do not have partial regulatory activity. They don't have one or two enhancers that are active, but that this is not enough to get expression of the gene. In contrast, in active cells, all the enhancers are strongly marked with the acetylation, and of course the gene is marked as well. So this is the first aspect of the activity, and at the Pitx1 locus, another aspect of the activity of the locus is change in 3D structure. So we also produce capture high C data set in here. And you can see here, and I will not describe this map in detail to this audience because I think it's pretty clear to everyone. What we can see is that in the GFP negative cells and the GFP positive cells, the structure overall is similar, but we see very strong differences. The first differences is that in the repressed GFP negative cells, we have this interaction between Pitx1, which is right here, and a gene that is located 300 kb away that is also repressed, that is neurog1. And in the active cells, we have a completely different structure where Pitx1 is establishing contact with all of its enhancers, whether it is the pen enhancer that we are studying, RA4 or other one. And you can see here with the subtraction map, the really strong difference in chromatin contact that you have between these two cell types. So we concluded here that the Pitx1 regulatory landscape appears to adopt the all or nothing activity. It means that it's impossible, or we don't see marks of activity of enhancer activity in transcriptionally repressed cells. Then we moved a bit further, and we saw that when we produce our fax profile, that there was actually two categories of active cells. One that is here with this first little hill of GFP cells, which is mildly expressing, not very strongly expressing the GFP, and the one here that are strongly expressing the GFP. And if we sort these two cells and we produce a K27 acetylation chip-seq on it, we can see that the lowly active cells have active mark on distal limb gene like OXA13 or EVX1, which is not the case of GFP, very strongly active GFP cells, which are depleted of these cells but marked with proximal marker. So again, here we are just able to sort proximal and distal part of the limb because proximal are highly active for Pitx1 and distal part are lowly active. And if we look at the Pitx1 profile, what we see is pretty interesting, because we don't see that lowly active and highly active cells use the same set of enhancer. We see that they use the same set of enhancer and not distinct enhancer repertoire. It seems that basic amount of acetylation over the cluster is over the gene and the enhancer is fine-tuned, but at a global level rather than at individual enhancer piece. You can see that a bit better at these different regions here. The only exception to that is this Pitx1 proximal promoter region that is right here at very close from Pitx1, which we are currently studying, and that seems to have very specific activity only in strongly active cell instruments. Right, so now let's move. What happens if we remove one of the enhancer? So if we remove one of the enhancer, first with the single cell data that we have here, what you can see is the expression of Pitx1. So and the cell density in a normal hind limb with single cell RNA-seq, we see that we have some cells which do not express Pitx1, and then a lot of cells that express it at different levels. If we remove the enhancer, we see something like that, like here in blue, with first a strong increase in cells that do not express Pitx1 at all, and a decrease in cells that are high expressors of Pitx1. It means that behind a very small change in average expression, we have actually a really strong shift in the distribution of expressing cells. And because, of course, single cell has a lot of caveats in terms of how deep we sequence every single cell, it was very fortunate here to have the fact sorting approach to be able to confirm this result. So if we take our fax, our GFP sensor here, and remove the pen enhancer, what we see is that globally the patterns are the same, with the exception that they seem a bit more blurry in the mutant. And if we fact sort these animals here, we see basically the same, which is here a strong accumulation of cells that do not express the GFP, neither Pitx1, and a loss of high expressing cells at the other edge of the distribution. So we really accumulate a lot of cells that are non-expressing. And to understand the phenotype, this is much easier than to just look at an average decrease. If a cell has 200 transcript of a given RNA and lose 30% of it, it's still a lot of transcript that are there and that provide the information of the cells to the cell. If now we accumulate many cells that do not have any of these transcript because the entire distribution has shifted, then we can start to understand how these cells can misbehave and produce a phenotype, a developmental phenotype. So we came back to the single cell and asked, where are these cells? What are these cells that do not express Pitx1 at all? And these cells are basically these two clusters here in the proximal part. And I show you here a violin plot where you can see that the irregular connective tissue here, or the proximal progenitor, have a pretty strong increase in the number of cells that do not express Pitx1 at all. And this has a direct impact into the proportion of these cells. So here in this graph, what you have is the proportion of each cluster in a normal forelimb and in a normal hind limb. You see, for example, the irregular connective tissue has around 12% of the cells of the limb are irregular connective tissue, and this is the same in the hind limb. And actually, it's the same between forelimb and hind limb for all clusters. Now, if we remove the enhancer, what we see is a decrease of irregular connective tissue and an increase of progenitor cells, which are basically these two clusters where we see indeed a very strong increase in Pitx1 non-expressing cells. So this is our link from a very weak loss of expression to a very clear phenotype at the end of the clubfoot. So the last point we wanted to address on that part here was to know what happened to these cells that stopped completely to express the Pitx1 gene. Is there something really fundamentally going wrong in terms of 3D structure, in terms of regulatory landscape activity? So what you can see here first is if we look in the GFP positive cells, right? So we know that in the wild type, they constitute 78% of the limb cells, but in the mutant, there are a bit less of them. In these cells, the removal of the pen enhancer didn't change anything in the distribution of acetylation over the landscape, didn't change the amount of transcript we have. It just reduced the number of cells, basically. And if we look at the 3D structure to compare both type of active cells, we can see that here the GFP plus wild type and the GFP plus without the enhancer look totally identical. If you look actually at this subtraction map, the only point that you will really see being lost is the interaction with the pen enhancer between Pitx1 and the pen enhancer here, which is logical because the pen enhancer has been deleted in a homozygous fashion in these animals. What we saw that was more surprising is when we looked at the GFP negative cells, right? So these are the cells that do not express GFP and do not express Pitx1 actually. And in these cells, in the wild type, as we saw before, there is no activity whatsoever at the enhancer or at Pitx1. But in the mutant, we accumulate a lot of cells that have activity and K27 acetylation at the Pitx1 promoter, as well as at several of its enhancer around. But this activity here is not enough to induce transcription, as you can see. There is no more transcription, or very marginally more transcription. So this is weird, because having this amount of acetylation over a promoter would tend to think that this is enough to induce transcription, but here it's not really the case. And actually, if we look at the 3D structure of these cells, we see that they are still in a repressed state. They are basically completely identical between GFP wild type, so GFP negative wild type and GFP negative mutant. The two cells are completely identical here. So this means here that if we think that the lack of active 3D folding, despite the presence of K27 acetylation, associated here with an absence of Pitx1 transcription. So what we think we have there is that normally the cells go from an inactive format with no 3D structure, active 3D structure and inactive enhancer, to a fully active structure and active enhancer. Here, we have just removed the enhancer and we have lost a couple of cells in the process, which were not able to change their 3D structure, despite the fact that some of their enhancer are active. And at the end, they could not manage to induce expression of the gene. So the conclusion here is the pen enhancer ensure robustness in establishing the active 3D structure of the locus across limb cell population. So the model for this first part is in the wild type hind limb, we have a lot of cells that express Pitx1 at different level. If we think only of the cell that have to express Pitx1 and not at the others, and when we remove the enhancer, we have a shift in the distribution, which change a little bit the average, but mostly made the appearance of these non-expressing cells that are basically active at the chromatin level and not at the 3D, and didn't produce a 3D chromatin change. These cells are mostly occurring in irregular connective tissue and proximal progenitors. And we think that they are really the reason for the phenotype, the clubfoot phenotype at the end. You can find more of this information on a preprint we recently posted. Now, after that, I will go to more preliminary data that we have on a completely inverted type of situation. It's actually pretty well confirming what we saw here, with the structure being necessary to induce transcription. And in this case, we are actually studying an effect that is called mechanism that is called regulatory endo-activation, that was discovered in several patients that show a arm-to-leg homeotic transformation, which is basically a transformation of the arm into something that looks like a leg. And what happens in this patient is that the pen enhancer that we were just talking about is inverted and comes in a closer proximity to Pitx1. And when that happens, Pitx1 gets activated ectopically in the forelimb of the mouse, but probably also of the embryonic human that have the mutation. And this is due here to a change in the structure, because not only Pitx1 comes closer to the pen enhancer, but also the entire 3D structure of the locus, here modeled by the Nicodemi lab, by the way, is changing. So you have normally in a wild type forelimb a repressed structure, where Pitx1 here in blue is in a domain with another repressed gene, neurog1, pretty far away from the enhancer, that are in a second domain and separated. In this inversion animals, what happens is that the structure is completely changed, and Pitx1 is now not close anymore to the neurog1 gene, which has been pushed away, but closer to the enhancer that are basically facing the gene, and as such produce transcription. So we wanted to know many things here. First, when we get this activation by one of Pitx1 own enhancer, do we completely recapitulate the normal expression, the normal activity that we should have in a hind limb, but this time in a forelimb? So we produced single cell data, and here I make a long story short, just showing you that in this forelimb, here mutant, we see appearing the expression of Pitx1 in many clusters of single cell clusters that are of course not expressing Pitx1 in the wild type hind limb, in the wild type forelimb. So these clusters that are expressing Pitx1 are actually identical to the cluster that express Pitx1 normally in the hind limb, right? And this is probably why, because this regulatory endo-activation keeps the cell-specific activity, that it produces a phenotype, right? And if you remember before, what I show you is the amount of cluster, and when we remove the enhancer, some cluster are lost. Well, when we ectopically activate the gene, some cluster and the same cluster that are lost normally in the loss of function are increased in proportion. So we really have here a reverse phenotype. Now, what happens at the transcriptional, at the 3D chromatin level? We can have a look at that by re-engineering the inversion in our sensor allele. So basically, we just re-engineer this. And if you look at the limb, you can see here that the forelimb, they start to have a little bit of signal in different cells. So you can pack sort the cell, and you can see that if you look at the GFP positive cells of the forelimb here, you see that you gain, of course, Pitx1 activity. So even in this context of a gain of function, our GFP sensor is able to detect the gain of function, and we are able to sort the cells where activity is going on. And in this particular cell, what is interesting is that we can see activity of, we can see acetylation of Pitx1, but nothing much else. I mean, a little bit of acetylation here, but they are, except for that strong acetylation at the promoter level, they are not so different from the GFP negative cells. For example, the PEN enhancer, which is right here, doesn't seem more acetylated in the cell where Pitx1 is active. So it's interesting that despite having similar regulatory environment, only some cells have managed to ectopically activate Pitx1, but not others. So we could think that it is the case, it is due to the fact that we have here acetylation on the promoter. But I just remind you what we saw before, that the GFP negative cells, when you remove the enhancer, also have acetylation over the promoter, but these cells here do not have transcription. So in summary, here we have a repressed Pitx1, which has no acetylation, an active Pitx1 transcriptionally that has acetylation here, and then repressed Pitx1 that has acetylation. So in this particular figure, we think the acetylation, of course, here is not enough to explain the expression of the gene. Something else must happen there. So in this context, the increase in Pitx1 promoter acetylation associate with transcription, unlike in the case of the loss of function. And if we look in these cells for the 3D structure, we have something pretty different than before that happens. If we look between GFP negative and GFP positive forelimb gain of function cells, you see here a change in 3D structure. It's pretty weak. It's weak because we have here a heterozygote allele, and we think that if we would have just a full homozygote allele, it would be a stronger effect, but it's still very clear. We can see that the GFP positive cells increases their contact with the different enhancers and decreases the contact with the repressed NROG1 gene, which is a clear feature of the activity here of Pitx1. So in that case, again, we see that the structure, when the structure change, we get transcription. We need, in a way, changes in structure to really get activation of the gene. So a transcriptionally inducing regulatory endo-activation associated with active 3D structure, despite the same trans environment. I would say here that we didn't change the content of transcription factors. Here we are looking at exactly the same transcription factor. We just changed the 3D organization of the locus, and because of that, we managed to get overexpression of the gene. So I'll just finish now by giving a few take-home message from that. The first one is that the Pitx1 regulatory landscape seems to display all not or nothing type of activity in the hind limb and regulate transcription level as a whole, not as a one enhancer plus the other, but that the entire landscape controls everything. The PEN enhancer robustly coordinate enhancer activity and 3D chromatin change. We saw that when we remove the enhancer, we are actually having enhancer that are active, but without a change in the structure, and therefore no transcriptional change. And final point here, the endo-activation can keep cell type specificity and has the opposite cellular phenotype as loss of function allele, which is expected, but still nice to see, and induce an active 3D structure and associated transcription, despite the same trans environment. So all of this could not be possible, of course, without my lab. And I would like here to thank especially Raquel and Olympia, who have been working on this project since 2 years. Antonella, Fabrice, Guillaume, and Rodrigue, which are members of the lab. And of course, like also to thank you for your attention and for inviting me today. And if I have some questions, I will be very happy.
So I was wondering, in the case where Pitx1 is expressed, you do see on your capture IC that you have a kind of contact between all the enhancers. Do you think these contacts are multi-way, or is just pairwise, and that's one enhancer will activate the gene one time and the other another time? Or you think it's a kind of collective effect that are responsible for the expression of Pitx1?
So yeah, it's a very, I like this question. It's very speculative from my side because I don't know. But in my opinion, we are here having, there is not 35 configuration that we saw so far. So my gut feeling here is that the locus can adopt two configuration. One is inactive and one that is active. So when it's active, probably there is a hub of contact with everything rather in proximity. And some of what we call enhancer are probably not enhancer. They are probably just due to their proximity with other enhancer and with the promoter of Pitx1 getting acetylation, just because they are close from the enzymes, they are close from, and so they get acetylation as a little bit as a collateral effect. And so I think it's really because there is this multi-way contact.
Okay, thanks.
But I have no way to prove it. Yeah, thank you.
Very cool.
Related to the part when you talk about the inversed locus with the pen and the H2AFY gene. So my question is how you can actually separate the two, I mean the phenotype of the two, because you have the two that are inverse, right? Would it be possible to do the same technique but keeping the gene in the right sense and only put the pen inversed?
It's a good point. Actually, this allele that I show is not the only allele that exists. There are many alleles where the deletion of the gene body of Pitx1 of H2FI produce the same effect, right? So of course, in that case, it's not a transcriptional effect. It's more like that you don't invert it, but you still lose the gene, right? In our case, the impact here is very local, right? We don't see anything else at the other loci. So, but is there a way to kind of remove, to do the inversion without changing this orientation? I don't know, actually.
So Ilya Flyemer, forgive me if I mispronounce your name, says, Guillaume, do you think all enhancers are active in each individual cell if it expresses PTX1, or do different cell types within the limb use different enhancers?
Okay, so this was what I was actually thinking at the beginning, that we would see something like that, but it seems that so far the enhancer, there is never one. We could not separate two distinct type of regulatory landscape that use different repertoire, right? So it's not impossible, but we would need to dig in another direction. So far, it seems that either you get everything active with some of the enhancer that are stronger than others that pop up earlier in the with lower amount of activity, and then you progressively add on top of that, but you never have like distinct sets. So I would say it seems that it's not the case, but so far we could not go further in that direction.
Mm-hmm. And they also had a second question, which is PTX1 and NeuroG1 both, are they both polycomb targets? And that's why they would interact in the off state.
This is what we think. Yes, they are actually both polycomb targets, and there is no other polycomb target in between. So this is why we think they are interacting here. Mm-hmm. Yeah, so we think it's kind of a common repression mechanism, but we could not go so far. We also even deleted the NeuroG1 gene to see if this was enough, for example, to release a part of the repression on Pitx1. This was not the case. But yeah, there is probably a repressive mechanism that is shared here between the two genes.
Mm-hmm. Then Marty Renom has a question. So Guillaume, thanks. My question regards the model of CTCF that might be helping enhancer scanning. Do you think something like that is happening here? Are any CTCF missing or methylated near the second enhancer site, such that cohesin does not stop enough time, if it doesn't pause enough time in most cells?
Yeah. Yes, CTCF definitely is helping here finding the enhancer. I would say there are many, at all the peaks that we see, there are CTCF sites. But the stuff is that there is no difference between the binding of CTCF or even of RAD21 and between the two tissue actually. So it probably provide a form of scaffold, which is then further, which is one of the possibility. And there is another mechanism that enables to take one or the other, but CTCF probably plays a role. We removed a few CTCF, didn't see anything big, but it would be interesting to be able to actually remove all CTCF of the locus and see what would remain. But I think it plays a role, but it's kind of a more scaffolding. It doesn't determine exactly the structure.
But there's no CTCF site that's deleted together with PEN in your PEN deletion?
In the PEN deletion, no. So in the PEN deletion, no, I think because the CTCF are a bit further out of the, I would need to check that. As far as I remember, they are outside of the deletion. We were careful about that, not to remove it, but I would still need to check.
Here I can slip in one of my questions. So I'm trying to think about, what's the model you have in your mind. So when you delete PEN, you don't affect, the ability to acetylate chromatin, right? So, but you affect the ability to form, to loop, right? To form this configuration. So what do you think is actually happening at PEN? It's binding of a sequence-specific transcription factor, I guess, that is not recruiting, co-activators, because co-activators might be getting them there, independently of PEN. So, but then how does PEN actually activate PTX?
It's a good point. So let's say the thing is more a probabilistic view. It means that if you have 100 cells and that can transform in a wild type when they have PEN, they can all transform into an active state. When you remove PEN, then suddenly you have, I don't know, 70%, 70 cells that can do it. What has happened in these 30 other cells, in my opinion, it's a question of you need a certain amount of activation to overcome the repression. And everything here happens probably simultaneously. You start to remove the polycomb, and if you go on the other side, and this I don't know exactly why, but obviously having more enhancer might help for that. And then you are in an active structure. So I think it's more like it's a balance between the repression and the activation. And here you kind of just, by removing that, you actually just shift the balance a bit on the other side.
So do you think if you did K27 trimethyl chip, you would see something that looks like a bivalent state? You would have signal K27 trimethyl?
If we could, because the problem in our negative cell population, where we have that, we have also a lot of cells there that are truly repressed, right? So we will see something that looks like bivalent, but we will need to do some sequential chip or something else to really show that really in the same cells we have that effect, right? Plus there might be an allelic stuff where one allele is still partially active, but yeah.
Yeah, good point. Okay, and there's one last question in the Q&A, again from Ilya Flyemer, who asks, are changes in 3D structure causal for activation? Couldn't it be that, 3D structure is a consequence of changes in gene transcription?
So I think in reality it probably co-occur. I mean, for now we have other data at other time where we see that change in histone marks seems during the decommissioning of a regulation to occur after the disassembling of the locus. So yeah, I tend to probably think that everything happens, one system reinforces the other. But what we would like to be able to do is to engineer the 3D structure of the locus in vivo, so to see if we can actually form some of the contact, and then see if by forming some of these contact ectopically, we first induce transcription and really then turn the locus completely around, even by just inducing one contact. It seems clear that if we do it that way, we will have, of course, if we bring an active enhancer close to the promoter, we will get activation. But in vivo, is that the primary event? It's a good question. I would tend to think that you need other things. For me, it's probably I probably think that there is like an effect that is kind of reinforcing. It's a reinforcing effect, but that the primary effect probably is likely to happen closer to the promoter without the need of 3D structure change. And then on the longer run, you start to have the folding because you find the other acetylated region, you find the other activators, and you bump, you make contact, and the contact then as such happens during the initiation phase when really you have the gene that starts to be highly activated. That's why it's difficult to actually separate both of them.